Hi, this is Anita Nag, and I am an assistant professor at University of South Carolina, Upstate. I'll be presenting a case study on glucose phosphate isomerase deficiency that I used as the end of the semester review for my Biochemistry 1 course. So often I find that at the end of the Biochemistry 1 course, students struggle with remembering what they have covered towards the beginning of the semester. For example, protein structure, function, pH, pKa, amino acid side chains. So one of the way I thought about handling that problem is using a case study that will incorporate all these concepts under the umbrella of one activity. For that, I'm using a case study which is used in medical and business schools frequently, but is used less in our biology and chemistry courses, especially for Biochemistry 1 course. There is very little time to do more case-based studies. During these case studies, what I found is glucose phosphate isomerase or phosphoglucoisomerase protein can be used to incorporate pH, pKa, amino acid side chains, different enzyme catalysis, enzyme mechanism, glycolysis, metabolic flask, all into one case study. So I've used patient data, clinical data published in Journal of Pediatric Hematology Oncology, and some of the research data that were identified on glucose phosphate isomerase and incorporated them into one activity. So this way we can connect multiple concepts under the umbrella of one protein. We can make connection between clinical data and biochemical observation. We can also encourage students to do more problem-based solving of any type of question, and then this way they can connect the question presented in the case study to the concept they have learned earlier in the semester and then apply those to solve the problem. And towards the end of the semester, we can also do a review this way and identify the concept that students are struggling with, and then they can study those for their final exam. We are covering several topics using this protein. We are covering water pH, isotonic, hypotonic, hypertonic solution, osmosis, pH, pKa. Under nucleic acid, we cover mRNA translation, DNA sequencing, the central dogma. Under carbohydrate chemistry, we do aldose, ketos, sugar ring opening, cyclization, reducing and non-reducing sugar. We cover almost all the basic concepts of protein structure function relationship, primary, secondary, tertiary, quaternary structure, purification techniques, especially gel electrophoresis, isoelectric focusing. And then under enzyme mechanism, Michaelis-Menten constant, Km, Vmax, doing Lineweaver-Burke plot, and then acid-base or nucleophilic catalytic mechanisms. And then finally, because this is glucose phosphate isomerase, we cover glycolysis, metabolic flask, and regulation of enzymes involved in glycolysis. So this is the structure of my course. Over the 15 or 16 week, we cover pH, pKa, amino acid, protein structure, purification during the first 1/3 when we take one quiz and exam. And then we move on to protein structure, function, enzymes, mechanism, kinetics. This is covered in exam 2. And then we move from there to lipids, membrane and transport, carbohydrate, glycolysis, part of glycolysis, which is covered by exam 3 and quiz 3. And then we finalize this with comparing glycolysis with gluconeogenesis and a very brief overview of nucleic acid. Now, before we finish the course, what I have been doing is giving the students an example of how to use case study to solve problem using a JChem 8 paper which dealt with glucose ring opening and A1C formation. And this is a great tool to cover several topics in one. And this was kind of an inspiration to develop the case study I'm presenting here. So once students get familiarized with how to work with the case study, we do our case study at the end of the semester, very last week, where we take two lectures. Each one is 1 hour 15 minutes. Students work in groups of 2 to solve this problem. Before doing the case study, they take a quiz, which is a pre-quiz, and it covers every topic. Very basic questions are asked. And we kind of evaluate how students are doing, remembering and retaining the material they learned. And then after they finish the case study, we do a post quiz, which are basically very same questions, a little bit modified. But we again see how doing the case study helps them retain the material better. And then they take a survey to give their feedback on the case study. I am going to show you 2 examples how we have structured this case study. So in the beginning, we give them kind of the story, and there is the story of this little girl, Emma, who had hemolytic anemia. And the doctors first found that her red blood cells are not actively going through glycolysis, but the major enzymes are fine. And then by some family history, they find that Emma's cousin, Sophie, had GPI deficiency. So then they actually find, okay, Emma also has GPI deficiency. So they do sequencing of Emma's DNA, and that's shown to the student. And they find out the sequence, applying their concept of Sanger sequencing, compared the 2 sequences, Emma's DNA and that of the normal individuals, and then find out using the codon table what mutation happened and how that actually changed one of the amino acids in the normal individual, which is arginine, to histidine in Emma's DNA. And then once they learn that, we tell them second part of the story, how Emma's red blood cells were sent to the biochem laboratory, and how we talk about the process, how they purified the protein. And students have to figure out why researchers were using water to burst open the cell, why they were using ammonium sulfate to precipitate. So they will have to figure out those steps. And then the researchers run 2 gels, native gel and SDS page. So students analyze these 2, and they can see that normally individual has 1 band in native gel that runs at 120 kilodalton, while in SDS page that is converted to 60 kilodalton. This is where students struggle the most to find out, okay, normal individuals have a dimeric protein. However, Emma has very tiny bit of dimeric protein. Most of Emma's proteins are monomeric. But once they find this difference, then they can actually, solve several problems. They can conclude what happens due to that mutation to proteins' primary, secondary, tertiary, and quaternary structure. They draw a hypothetical gel of isoelectric focusing. Since there was an arginine to histidine mutation, they can draw the hypothetical gel. And then once they finish this part, they're again given another set of story where they're given substrate concentration versus Vmax for EMA's GPI, and they calculate the Km, and then they are told that certain intermediates are found in higher level in EMA's red blood cells, and they identify that because glucose 6 phosphate is accumulating due to the deficiency of GPI, in EMA's red blood cells, pentose phosphate pathway is upregulated, and that's why some of the intermediates that are accumulating, they can figure that out, making those connections. As we carry on with this case study, students keep on making a list of topics they are covering. This is an example of that. So they have written glycolysis, regulation of glycolysis, pentose phosphate pathway, metabolic flask, enzyme catalysis, and then they identified the topics they are struggling with and make a list of them so that they can study for their final exam. This is one of the questions from the survey. We asked them how much confidence they had retaining the material before they entered into this activity. And most of them say that they have some level of confidence they will retain this material 3 months from finishing the semester. Once they did the case study, they now feel more confidence, strong to moderate confidence that they will retain this material 3 months after the case study. So this is a huge improvement of at least student confidence that they can relate some of these materials to a real life scenario. They can see how the clinical data can be applied, how that can be used to combine with research data to solve a problem. And many of our students are pre-med students, so that kind of helps them to actually appreciate biochemistry even more. And at the end, I'd like to thank two of my colleagues, Dr. Linnea Freeman. She at Furman University applied this case study to her class as well and gave me some feedback. And Dr. Selena Cush's office at USC Upstate, who helped me do the survey. And then finally, this is a picture from my class. Students gave me permission to share this. They are working in group of 2, and as you can see, they have all their books and notes open. They are very engaged in this. It's almost like a competition among these groups who can solve this puzzle. So students were very excited to do this case study. And finally, thank you all for listening, and ASBMB for this opportunity to present our work.